Assalamu alaikum class. Today we are going to study about immunology. This is the general overview of the second section of course. By the end of this lecture, you will be able to understand immunology, immunity, susceptibility, antigens, and antibodies. You'll get an understanding of the importance of immune system and what happens when we lack it or it goes wrong. You will be able to differentiate between innate and adaptive immunity and identify different types of adaptive immunities and how they are acquired. Immunology is the study of components and function of immune system, how immune system help us protect against foreign invaders, and how this information can be used for better diagnosis, prevention, and treatment of diseases. Now let's see what an immune system is. The system is composed of molecules, cells, tissues, and organs which are present throughout the body, which provide both non-specific and specific protections against foreign invaders, which may be microbes, their toxin, or tumor cells. Because human body provides an ideal environment for many microbes, they try to break in. It is our immune system job to keep them out, failing that to seek out and destroy them. That's why it's crucial for human survival. The term immunity is derived from a Latin word, the immunitas, which refers to protection from a legal prosecution offered to Roman senators during their tenure in office. Historically, immunity means protection from an infectious disease. A person who is protected from an infectious disease is called immune. A state of being non-protected is called susceptibility. If a person is not able to defend itself from a specific microbe, he is said to be susceptible to that specific pathogen. Whenever a foreign invader gets entry into our body, the immune system detects it, deflects it, or destroys it. So the overall collective and coordinated response to a foreign invader is called an immune response. There are two types of immune response, either being non-specific or innate response, or specific or adaptive response. We will discuss them in detail in the further slides. But you can see there are three lines of defense which has to be breached by the pathogen to affect our normal function of the body or to cause disease. The first line of defense is non-specific, composed of mechanical, chemical barriers and our reflexes. The second line of defense is also non-specific, which consists of inflammation, fever, phagocytosis, and action of natural killer cells and cytokines. The third line of defense is basically the adaptive immune response, which is highly specific and it is either cell mediated or humoral, meaning by means of antibodies. So the pathogen has to reach all these layers to cause an infection. So the primary function of immune system is to defend against infectious microbes, which can be bacteria, viruses, parasites, fungi. And these anything which can trigger an immune response is called an antigen. But there are some non-infectious foreign substances who can also elicit an immune response by producing antigen-specific proteins called antibodies. Furthermore, the mechanism that normally protects individual from infection and eliminates foreign substances can also cause tissue injury and disease in some situations. So whenever a foreign invader gains entry into the body, the immune system checks it through several checkpoints. First, it checks whether invading material is self or non-self. If it is self, there is no immune response generated, and if it is non-self, it has to pass through another checkpoint. And it's check whether the non-self substance or the microbe or invader is either harmful or not to the human body. If it is not harmful, it can again result in no response. However, if the non-self substance is declared harmful, it will trigger an immune response and the immune system will try to eliminate it. The immune system also has the ability to identify cancerous cells or damaged cells and result in elimination of these cells as well. So there are two functions: defense against infectious microbe and removal of tumor cells. Historians often give credit to Thucydide in the 5th century of BC in Athens as having first mentioned the word immunity to an infection. He wrote in his book that persons who have been exposed to plague previously could care for the sick without danger. That's when they are protected from the plague. The concept of protection immunity may have existed long before, as suggested by ancient Chinese custom of making children resistant to smallpox by having them inhale the powder made from skin lesion of patient recovered from the disease, process called variolation. As you can see in this picture, first vaccine was developed by Edward Jenner in 1798, and we have discussed in our first lecture of this course. And in 1890, Waring and Catasto developed the first antitoxin for treatment of diphtheria. Thanks to the knowledge of immunology, in 1980, smallpox was the first disease that was eradicated worldwide through a program of vaccination, according to WHO. As you can see from this graph, the number of cases of smallpox from 1965 reduced gradually to 1975, and finally in 1980 they were eradicated thanks to the vaccination program. These are a list of some of the diseases which have been controlled by use of vaccines. As you can see, the percentage change is ranging from 87 to 99.9% decrease in the disease due to vaccination program. Since 1960s, there has been a remarkable transformation in our understanding of immune system and its function. Advances in cell culture techniques, including monoclonal antibodies production, immunochemistry, recombinant DNA methodology, and X-ray crystallography, and the creation of genetically altered organisms, especially the transgenic and knockout mice, have changed immunology from largely descriptive science into experimental one, in which diverse immune phenomena can be explained in structural and biochemical terms. So why is immune system important and what happens when we lack it? Let's see with the help of example. It's a boy called Bubble Boy. His actual name was David Vetter. He was born with a severe combined immune deficiency called SCID, which is one of the most severe type of primary immunodeficiencies diseases. At the time of birth, 1971, a bone marrow transplant was the only cure with an exact match. But unfortunately, there was no exact match at that time. So he had to live in a very protected environment created in a bubble. That's why he was called a bubble boy. So this environment was germ free and he lived in there for about 12 years at Texas Children's Hospital. At the age of 12 years, he eventually received a bone marrow transplant from his sister. And although his body didn't reject the transplant, he became ill and developed lymphoma. An autopsy revealed that Her sister transplanted bone marrow contains traces of dormant virus called Epstein-Barr, which at that time was not detectable by pre-transplant treatment, and the virus was probably responsible for David's lymphoma and his death. So you can see how much immune system is important for our survival in a normal environment. Now let's see what happens when our immune system goes wrong. Sometimes what happens, the immune system checkpoint which differentiate between harmful and non-harmful stop functioning. In that case, the immune system responds to a seemingly harmless foreign substance such as pollens and lead to allergies. So in that case, the antigen is called an allergen. A second abnormal situation arises when the immune system can mistake self for non-self and launch an attack against body's own cells or tissue. The result is called autoimmune diseases. Some of these diseases are listed here, for example, rheumatoid arthritis, GBS, diabetes type 1. In autoimmune diseases, the body produces autoantibodies. These autoantibodies attack healthy cells and result in damage. For example, in type 1 diabetes, autoantibodies damage the pancreas. The study of immunology helps in different fields of medicine like tissue transplant, diagnosis of disease, prevention of disease via vaccination, blood transfusion and pregnancy immunology, and treatment of cancer via immunotherapy. So students, it's time for our first concept checkpoint. First question is you have to differentiate between an antigen and an allergen. Please write one example for each. And second, you have to differentiate between antibodies and autoantibodies. Again, you have to give one example. Second question is why polio is still not eradicated from the world despite WHO vaccination program in many countries. Third, discuss the role of immunology and its uses in fighting COVID-19 pandemic. Try to answer these questions. We will discuss the answer in our interactive section. Defense against microbes is mediated by early reactions of innate immunity and the later response by adaptive immunity. So innate immunity is also called natural or native immunity, which provides the early line of defense against a microorganism or an antigen. This defense mechanism is present since birth, before infection, and respond very rapidly to any infection. The mechanism of innate immunity is not highly specific. That is, it is not able to distinguish between very close related microbes because it targets only the features which are common in related microbes. This type of response has no memory. That means this mechanism reacts only to microbes or its product, and they respond in essentially the same way regardless how many times it is exposed to the same microbe. So the response would be the same in every exposure. This type of defense mechanism is attained by various barriers like anatomical barriers, skin and mucosal lining, physiological barrier like pH and temperature, presence of some phagocytic cells forming the phagocytic barriers, and inflammatory barriers which causes inflammation and prevent the spread of infection to other places of the body. Innate immune responses presents immunity's early warning system and are designed to prevent microbes from gaining access into the host body and to help eliminate those that do gain access.